to introduce today's IGB seminar speaker, who is Sushmita Roy, who's coming from the University of Wisconsin at Madison, where she runs a computational biology lab using statistical machine learning methods to understand gene regulatory networks. And she's going to be telling us a lot about their recent work on gene regulatory networks, especially using single cell data, which I know is something that many of us here are really interested in. She's also done some really important work on the evolution of gene regulatory networks and is increasingly inferring GRNs from single cell data. Sushmita is an associate professor in the Department of Biostatistics and Medical Informatics, as well as the Department of Computer Science. So I think that her talk is going to be resonating with the wide, diverse IGB community. So with that, I will turn it over. Oh, and I've also been encouraged to mention that if you have any questions during the talk, Dr. Roy is welcome to be interrupted.
All right. Everybody can hear me at the back? Awesome. Thank you, Allison, for that very nice introduction, and thank you for inviting me to come here and tell you about some of our work. So as Allison mentioned, our group focuses on the development and application of machine learning methods to look at different problems in gene regulation and gene regulatory networks. So at a high level, we are interested in three major directions. So we are interested in developing computational methods that can be used to infer genome-scale gene regulatory networks, and I'm listing some of these tools over there. And we are also interested in understanding a 3D genome organization, and that's something that I won't get into today, but that's another level of regulation of gene expression, and we've developed tools to look at different computational tasks in understanding the 3D genome organization across different conditions. And then the third part is we are interested in looking at regulation of gene expression and gene regulatory networks across long time scales, evolutionary time scales. So this is the gene regulation theme, so you probably know what I mean by gene regulatory networks, but I just wanted to just make sure we are talking about the same thing. So a gene regulatory network is basically a network that establishes connections between transcription factors and target genes, and we like to depict them as directed graphs. And we are so interested, and we and the community are so interested in defining these gene regulatory networks is because we think of them as the control machinery in cells. So they specify what sets of genes get expressed when and where. What we've been recently focusing on is regulatory networks in cell fate specification. And so this is a Waddington landscape that you might have seen, and here are cells in different states, and we know that there are underlying gene regulatory networks that drive these different transitions. But the challenge is that we don't know what these gene regulatory networks are. And so in today's talk, what I want to tell you is some computational tools that we are developing from bulk and single cell omic data sets to try to get to some aspects of this question. So the first part, the shorter part of my talk, will focus on some tool that we've developed to look at multi-omic bulk data. And the second part, which is more, has actually more parts to it, is focusing on single cell data. Okay, so let's again think about what do we mean by a regulatory interaction between a regulator and a gene. It's useful to think of it in two aspects. And it depends on where you're coming from. So some people think of basically what I think of as cis interactions as a gene regulatory network. So when we think of a gene and what regulates its gene expression, there are regulatory sequences which are in the promoter of a gene, and also that can be far away from the gene where transcription factors bind. And it's so basically the expression of this gene is largely regulated by the sequence composition and who binds to regulate the expression of the gene. And so I call them as cis regulatory networks. And then there are these trans regulatory networks, which are where we are specifying what transcription factors levels activate the expression of the gene. So it's useful to think of both of these types of things. And here we may think of things that may not directly bind to DNA. We may think of things like signaling proteins and other more upstream types of things. So, and with technology today, we can actually measure many different aspects of gene regulation. And this is a picture from the ENCODE consortium where one of the goals was to try to define the compendia, what are the regulatory elements in the human genome. And so there are a lot of different ways in which we can try to assay or figure out what are the regulatory components of in the genome. And what has happened now with bulk data is that we have a lot of many different types of these modalities. So we have ATAC-seq data, we have chromatin data, we have, of course, gene expression. And so just to give you a sense, here are 4 papers from the last few years where the question is to look at a dynamic system. So the first one is looking at cellular reprogramming, and this is differentiation, where people have collected a lot of deep data, so lots of histone modifications, accessibility, and the goal is to try to infer a gene regulatory network. So, but this is a hard problem, and the reason why this is hard is because there are a lot of things that we are measuring, but we are not measuring a lot of samples. So typically when we are trying to infer gene regulatory networks, we are saying, okay, I want to establish an edge between a regulator and a target. I need to observe a large number of samples to be able to establish those relationships. And so the issue is that you have a lot of variables that you're measuring. There are too few samples. And the third thing is the notion of relatedness. So these are dynamic systems. So there's a time series or there's some kind of a lineage structure. And we ultimately want to be able to capture those dynamics. So how do we try to address that problem? And one thing that I'll already say from upfront is, so we love trees. And so the trees can be phylogeny, lineages or something, but a lot of work that we do, we tend to think about how things are related, and trees, of course, capture those relationships. So you'll see this theme of trees a lot in my talk. Okay. So the problem is, so we have bulk data sets that have a lot of different types of things that we are measuring, and we want to be able to say something about dynamics of gene regulations. How do we do that? So we developed a tool called Dynamic Regulatory Module Networks, or DRMN for short, which gives you one way to look at this problem. And so the input to this is some kind of a relatedness. So this can be a cell lineage, and then multiple types of measurements, one for each cell type. So it can be gene expression is required, and then the other are optional, although we think that accessibility is, at least if you have accessibility, that would be great. And then we have a set of sequence-specific motifs, and then as output, we produce for every cell state, or for every cell type or every time point, we produce what are called regulatory modules. So we don't have enough data to actually capture fine-grained per gene regulatory networks, but what we can do is we can group genes into sets and then try to infer regulatory information at the level of groups of genes or sets of genes. So just to give you a little bit more detail, so let's say that we have one cell type. What we are trying to produce are basically groupings of genes into expression states or modules. And for each of those, we are also inferring what are the regulators. So in this case, we are showing two sets of genes that are in the high state and the low state. And what this regulatory module network is trying to do is predict what are the cis elements that are in these gene promoters that are driving the expression of these genes. So that's the prediction problem. But we have multiple, actually, time points and cell types. So what we are trying to do is we are trying to learn these models in a joint learning framework. So the framework leverages the fact that these cell types are related. And that helps to infer more believable dynamics across this system. So the way we do this is by putting it in a multitask learning framework where each task is the problem of inferring these parameters that relate the regulators to the expression states. Okay? So we have applied this to a bunch of different types of studies, and here I'm listing the types of modalities that were available. So all of them have to have gene expression, but the number of samples is very few, and some of them just have just accessibility information. So we've used all of this together to try to infer some regulatory information, and I'm going to tell you what I mean by that. So the system the data set that that I'm going to tell you more about is this one, which is studying cellular reprogramming, that is going from these mouse embryonic fibroblasts to an induced pluripotent state, and a whole bunch of different things were measured. So we are not going to use these transcription factor ChIP-seq. We are just going to use the histone modifications and gene expression and accessibility. So here is what we get. So this is these are the 4 cell types, and these were linear. So this is like a linear relationship across these samples. And for each of these little bars corresponds to a set of genes that are in a particular expression state. So these are in the lowest state, and these are in the highest state. So there are 7 of those. So we are discretizing the gene expression into these different 7 states, but we are doing this jointly across all of the different cell types or time points. And then the important thing is that we don't assume that the genes within each of these states are the same. So the question is, how much do they change? And so these are telling us how much do the genes actually change across the different modules. And from here, you can already, from here you can already try to get a sense of large-scale dynamics. So what we see is that so each of these numbers corresponds to how similar are the genes that I identify in these set of modules to these set of modules. Actually, this per module. So there are 7 of these, and this is globally how similar they are. And so what we see is that largely the MEF and the MEF48 are very similar, and then these 2 are very similar, but there have been major changes between these 2, which makes sense because this is where largely there are a lot of transcriptional changes that might be happening. Beyond that, so this is all at the gene expression, like at the module level. What are the regulators? So what you're seeing here are the key regulators associated with the 3 highest expression modules, and we see histone modifications as well as different transcription factors, and many of them make sense, like ESRRB. I can't actually see over there, but some of them are known to be associated with the establishment of cell state. Okay, so that's so what we've done is we've basically taken our gene expression data and we've grouped those genes into different states, and we've also predicted what are the main regulators that drive these different expression states. We can also get more fine-grained dynamics. And the way we do that is by looking at individual genes. So this is the NANOG gene, and you might know this, but NANOG is one of the key genes that needs to be upregulated in the embryonic stem cell state. And what we can do is, once we've run DRMN on our data, we can look for genes that change their state from one state from one cell type or one time point to another. So in this case, the nanoc gene is going from the low state to a high state, and these are the module assignments, and these are the actual gene expression levels. And we can get more genes that actually exhibit that, and furthermore, we can find regulators that we have predicted at the module level to further, so we learn another regression model once we've done that, to find regulators that change together to explain the expression of this gene. So the way we want to be able to get individual gene level regulatory information, but if you do it in a naive way, you don't have enough data. But what we do is by grouping the data first into different discrete categories, then identifying what genes change and just zooming into those genes and trying to find cis regulators that drive the expression, we can get some interesting pieces of information. And so we've done this across a bunch of different systems, a lot of mammalian systems, but also plants. I just want to tell you quickly about this other collaboration that we had with Matthias Kirst and Jean-Michel Ane. He's at UW-Madison. Matthias is at University of Florida, where the system here we are looking at is nodulation in this plant called Medicago. And here we were interested in looking at the dynamics of this process of how the symbiotic relationships is established between root nodules and symbiotic. So these were like chemicals that simulate what signals a bacteria produces. And so we had RNA-seq data and attack-seq data, and we applied our framework where we did a bunch of different analyses, including DRMN. We inferred gene networks, and we inferred gene modules, and so on. But we used this to try to prioritize regulators, and one of the nice things that was done was also validation of some of these regulators, where they were knocked down and then measured their impact on phenotype, and we found 2 of the 3 regulators that we predicted were also had a valid phenotype. So that summarizes the first part of my talk, which largely focused on bulk multi-omic data. The challenge with these data is that we don't have enough samples. You have a lot of deep measurements, but we want to get to GRNs and we want to get to temporal dynamics. So how do we do that? And we do that by incorporating this within a joint learning or a multitask learning framework, and we group genes into modules or sets, and then try to leverage this clustering type of an idea to try to infer these relationships. And we've basically applied this to a reprogramming system and also in a symbiotic system, and we found some interesting results. Questions for the first part of my talk? Okay, we can move. Oh, yeah, questions.
So the multitask portion, I saw there's 2 portions. It looked like one was inference of the
Modules, right.
Yeah, what's the second one?
So the regression to find the regulators. So the multitask is only coming in the first part of the model, which is the main DRMN output. So in order to learn the regulators for each of the cell types or time points, that's where we are solving regression problems jointly within a multitask learning framework.
Yeah, so is that right? There's 2 tasks?
Oh, so there are as many tasks as there are cell types or time points. And you're solving, for each of those, you're solving a regression problem that tries to link regulators to groups of genes. But then once you have grouped those genes, there is another regression, and we actually do it in 2 ways. It's more like a post-processing step. It depends upon the number of samples that we have. But for a group of genes that is dynamically changing, because all of the DRMN thing that it's doing is it's giving you at the expression state level. It's not giving you at an individual gene level what the regulators are. To solve that problem, there we are using another regression framework, but the type that we use depends upon the number of samples. So if you have like very few samples, then what we do is we actually just flatten out the vector and just do a single regression. But if you have more samples, like 8 or 10 or 20, then we do something based on group lasso. So Okay, so that was all bulk. We will now move to single cell, and I hope I have time to cover all of the things that I wanted to tell you, but anyway. So here we are really diving into single cell, really leveraging the single cell technology to get into cell type specific networks. And I don't have to tell this audience, but this is really revolutionizing biology. And there are so many different types of modalities being used for many different questions. And so there's a lot of excitement in thinking about these types of data. And we've been all very excited about this because they give us a whole new way to think about gene regulatory networks and cell type specific gene regulatory networks. So given single-cell RNA-seq data, let's say, or single-cell omic data, what are the questions that we need to solve to try to define gene regulatory networks on a cell lineage? So the first question is to define what are the cell types. The second question is how are they related? So that's the structure of the lineage structure. We want to know what the GRNs are for each of these cell types. And we want to know how they change, to look at dynamics. And then finally, we want to know how correct are they. And this is a hard question. I will not get into it, but I feel like this one person cannot, or one lab cannot solve this problem. But as a team, maybe we can solve it. But I'll not get into it, but we can talk about some ways to maybe evaluate GRNs. Okay, so let's look at the first question of what are the cell types. So here's a very common workflow of analysis of single-cell RNA-seq data. You start with a matrix, which you have cells along the rows and genes along the columns. And then we do some dimensionality reduction, some clustering, and then annotation based on known marker lists or cell atlases. And there are a lot of methods that are out there. Among some of the methods that we've seen have a lot of leverage is something called matrix factorization or non-negative matrix factorization. So the data that we have are non-negative, so they're all like zeros, so the count data, and the data are sparse. So what NMF gives you is a way to give you a low-dimensional representation of your sparse gene expression matrix. And the way it does this is by approximating this single-cell RNA-seq matrix as a product of two low-dimensional matrices, U and V, and what NMF does is basically tries to solve this optimization problem where you're trying to figure out what the best construction is to minimize the error between the original data and the reconstructed data. But what is nice from NMF is that you can, it is not only a dimensionality reduction problem, not only a dimensionality reduction tool, it is also can be used for clustering. And you can use these factors. You can use, you can actually group the genes and the genes and the cells based on how likely is each of the factors contributing to each gene's expression or each cell's overall expression, and you can use that to extract out groupings of cells and genes. So here you can basically take this matrix and say, oh, these cells belong to cluster 1 because factor 1 contributes the most to this cell's expression, and so on and so forth. You can get out these factors, and correspondingly you can get out genes. And that's what is the nice thing about NMFs. It's a biclustering problem, a biclustering approach that will give you cell clusters as well as genes that go along with it. It doesn't solve the problem completely, but it gives you a place to start to get you potentially good gene markers. And we've applied this in a particular study where we collaborated with Randy Ashton. He's a biomedical engineer, and his then postdoc, now an assistant professor, and a scientist in my group, where we looked at very early hindbrain development in human, and we found these different cell clusters. Many of the cell types that we identified corresponded to known cell types, but also new cell types that we didn't know exactly what they were. So this was applying NMF to one data set, but what is happening now is that typically you have single-cell RNA-seq data for multiple samples. So how do we handle a multi-sample data set? And so many of the existing approaches that have been developed tend to assume that all the data can be projected into a single low-dimensional space. And that may work, but in many situations, that may be a very strong assumption. And the other thing that was shown, so this is really a huge and exciting problem. A lot of people are working on it, and there are a lot of methods out there, and also very systematic benchmarking studies. So this was a study from Fabian Thies's group where they looked at a whole bunch of methods to try to integrate data across different platforms and omic levels and everything. But one of the things that they found was that it remains challenging to actually integrate data across multiple species. So here is where I'll actually talk a little bit about multiple species from the evolutionary side, but most of my talk is actually within species. So what we've done is try to, and we are very interested in actually looking at single cell data across species at the cluster level, at the cell type level, at the GRN level, from the single cell, also from a bulk point of view. And the way we are looking at this problem is by using matrix factorization. And this is why I introduced to you non-negative matrix factorization. So NMF, or non-negative matrix factorization, has been used to integrate multi-sample data in two, I would say, popular ways. In one approach, what people have done is, so the genes are the same across all those different conditions, across the different data sets. So you can stack up the matrix and then apply a single NMF, and that will do some integration for you. Okay, so that's one way to do it. The other way is based on Liger, which some of you might have heard. And the idea here is to do two separate factorizations, but share a component, which is the black little matrix over here, to on the gene space. So what you're doing is that you are getting data set specific factors as well as a shared space, and that can be used for doing data integration. So Liger is a pretty good method, but we find that it is a little bit restrictive to assume that only this is the shared aspect of the data set. And it does it basically, if you have 10 data sets, it will find one shared space across all the 10 data sets, so which can be a little bit rigid. So what we've done, again, here comes trees again. So we've tried to actually take a different way look to handle this problem. And so what we're saying is, let's say that you have 3 data sets, 1, 2, and 3, and it is pretty reasonable to assume that these data sets that are maybe differently related, to put them on some kind of a hierarchy. So these 2 data sets are more similar to each other compared to this third data set. And what we do is we apply NMF to each of these data sets, but then again we do this in a multitask setting. And the way we do this is by specifying this objective where we are saying that our expression matrix, so this is the standard NMF objective, but what we are saying is that these factors, V, the gene factors, they should be constrained such that those data sets that are close to each other should have more similar low dimensional representations than the data sets that are further away. Okay, so that's the general idea, and we can control how much we want this to actually influence our overall solutions by tweaking this hyperparameter over here. Oops, sorry. Yes, question. Big A is the set of, okay. Right. So big A is the set of edges that are there on this tree. So yeah, basically all things that don't have a root. Is that clear?
Yeah, I think so.
Yeah. So basically for every V, so we infer a V in every point in the tree, and we want the V's for every non-root node in the tree to look similar to its immediate parent. To its parent, not immediate parent. Yeah. Okay. So we have applied and compared this approach to a bunch of other methods. So here you can see is TMF, which is our method, which is called tree-guided matrix factorization, Liger, Scanorama, Harmony. Joint NMF is the approach where we have a concatenating things together in on one direction. And then multi-view NMF, which is actually, I would say, TMF is a more general version of multi-view NMF. So multi-view NMF would be where you have one flat tree. So all the data sets get, the factorizations look similar to one root matrix. And I think it was developed by, well, there are some variations of it, but at least one variation of it was developed by somebody at UIUC. Anyway, okay, so not for single cell, but much earlier. So we have looked at how well, so we've done a benchmarking study, and the purpose of this is to try to tell you that, TMF is a reasonable method to use, and we've looked at it from a clustering point of view, and also batch effect correction, and how well it integrates the cells across different data sets. And what we see is, this is for a pairwise data set. It's actually the famous PBMC data set that a lot of people use for benchmarking. So our method, which is in green, is generally ranking at 1 or 2. Then we looked at a more complex data set where we had lots of samples. So not lots, we had 9 samples, but it was complex in the sense that it was coming from multiple lines. So these are organoid data and multiple and 2 time points. And so here also with this complex structure, we are able to do reasonably well across different metrics. And then we have now also looked at multiple species data where, so this is from an organoid data, and this is actually from brain from different animals. And we find that across these different metrics, TMF is a good method. And it's hard to actually find the best method. So typically, different methods have different strengths and weaknesses, and so it's almost always good to use maybe 1 or 2 methods rather than tying yourself to one method. Okay, so now a little bit about what we found. So this is this data set from Canton et al., and here they were studying early brain development, and so they have organoids from these 3 species. And you can look at the number of cells that they have. And so the macaque data set is quite small, but there's a lot of heterogeneity across these data sets. So one challenge across species, right now we haven't solved it, but we are working on it, is this notion of gene duplications. So right now we are assuming that either the gene is there, or it's conserved, or it's lost. So there'll just be zeros in our matrix, which is okay. And so for the losses. Okay, so this is what it looks like. This is a UMAP that kind of shows how the different data are laid out. It's just to dig in more into the results. So what we see, so we looked at the results from these 4 methods: TMF, Liger, Scanorama, and Harmony. And we can see that Harmony and Scanorama tend to find less conserved clusters. So that's what we really want to do across these different data sets. We want to find a method that is able to find more conservation, because conservation is easy to interpret, because if things are divergent, that becomes a little bit challenging. Of course, you're interested in divergence as well, but we want to make sure that at least the conserved signals are being captured well. And we see that TMF and Liger, so TMF is slightly better than Liger, where we see more conserved clusters. So yeah, that's what you see across these different data sets. And using TMF, so TMF internally is using NMF, so it also gives you gene markers associated with the different cell clusters. And with that, what we did was we looked at literature to see what the genes are are, and we can annotate some of our cell clusters. So here are different annotations. This is one particular cell cluster, which is which you call the neural stem cell, neuroepithelial cell type, and you can see what the cell clusters are and the distribution across the different species. And then we can find marker genes that are associated with them. So this particular gene, HES1, its expression is shown in each of these different UMAPs. And we can see that the HES1 is overlapping the cell clusters. It's a nice marker gene. And we've done this for the other cell clusters as well. And these are different cell types. And we've identified different marker genes that are associated. So the TMF markers that we get out, so it does a reasonable job of data integration. It finds these cell types and also identifies marker genes that are useful to try to annotate these different cell clusters. And then finally, we've also done Go enrichment to further validate what the clusters mean. And many of these processes make sense in terms of the annotations that we are assigning them. So this is the first part of the second part of my presentation on single cell data. And so the overall goal of TMF is to try to combine data from multiple conditions or multiple species, multiple treatments, and try to do dimensionality reduction to infer cell clusters and also annotate cell clusters based on the genes. And our initial analysis here is on organoid data. This is publicly available data, but we are also in collaboration with people where we are also generating single-cell RNA-seq data, but we are happy to apply our approach to data sets that might be you think might be a good example for this method. All right, questions for this? Yes.
Can you go back to the mathematical formulation of TMF? Yeah. Okay, so I have a couple questions. I guess my first is in the results, how did you choose the similarity alpha?
Good question. That's a hyperparameter. So we tried a bunch of different alphas, and then we went with what looked like a good integration score for our method. So if you make alpha very small, then you will not get very shared genes. What we've seen is across different data sets, generally an alpha of 100 is a reasonable value.
So just out of curiosity, so if you make alpha really big, you're going to get excellent integration, right? Everything will look the same. So how did you balance?
Yeah, so I mean, so we want to get cell type specific. I think it'll not like completely, integrate. So there, at some point you don't want this term to overcompete like this. You ultimately want to make sure that the loss is the one that's dominating the data. The cell clusters, so if you make it too high, the genes will be very, very similar, but the cell clusters don't look good anymore. So it's like a trade-off between how good the cell clusters look and the genes that are driving those cell clusters.
My second question is, you were comparing this to a bunch of other methods for either clustering or integration, but those could be nonlinear, and this is linear, I guess. Did you notice?
Yeah.
There was, I don't know, did that matter, especially for the clustering part?
Yeah. So all of these methods are, I would say, so Scanorama, these are all matrix factorization. Internally, they're doing some sort of matrix factorization. Harmony is actually doing PCA and then trying to do some cleanup after PCA. So in that sense, these methods are largely in the same family. We didn't compare to other methods like SCVI, which is like a deep method, which I would say is more nonlinear. And I don't know how well it compares to SCVI. I think SCVI is actually a pretty good method. So if you have GPU, use GPU.
And then did you need to wait for different numbers of cells in your different views?
Did we have to wait?
Yeah. So if you look at the loss function, the first term is just treating, it seems like it's treating all the Frobenius norms from the different L's the same, but did you feel like it made a difference if you weighted by, let's say, the number of cells in a particular view or a particular species?
We didn't do that, but that's a good, that's something to think about. Also maybe downsample, but we don't want to throw away the data. But that might be something to consider because our data are very heterogeneous. But each of those are actually separate factorization. So yeah, you're wondering like, with the sharing, will the human data dominate all of the results or not? Yeah, we didn't look at that. Yeah, it's a question at the back.
Do you have any nice evolutionary stories which you learned from comparing the chimpanzee, macaque, and human brain? Do you see any sort of de novo modules which are only present in human chimpanzee?
We are still looking at the results right now, and I don't have very good, stories, but we are trying to look at our results now and trying to see what we are actually learning. And one thing what we see is there are like some clusters out of the 20 there are. So the blue, I should have explained a little bit more. The blue is where the cluster is less present in that species, whereas red is present across. And we see there are of course, macaque tends to have a lot of clusters that are not there. So a lot of clusters that are there in human and chimp. But we've not, so right now we are at a stage at which we are looking at genes and how they are changing, and we should have more biology, hopefully, in the full paper. This is still work in progress, yeah. Okay, I do have stories in the last part of my talk, but I probably don't have time, but not evolutionary, unfortunately. Okay, so the last part is really focusing on gene regulatory network inference. And both what I'm going to talk about is in this reprogramming system. And again, this is just a recap of what I said before. We have to think about cis and trans. And this is a collaboration with, and this is why I had reprogramming in the first part of my talk, because Rupa, my very close collaborator, studies this reprogramming system. And for this system, this is her student, my students, and we had RNA-seq data, and now we have attack-seq data across time for the same system. And what you're seeing here is what the attack data looks like. Here we just use Archer, and this is just to make sure that the data are reasonable, and we find tight clusters associated with embryonic stem cell state as well as the starting state, and they form two distinct clusters. And we find that there are gene markers that are driving them, and so this is where you see OCT4 or POW5F1 that is highly expressed in the ES, which makes sense, and then MEFS has this muscle-related genes. So overall, sanity checks, this looks, Archer gives us reasonable results, and our data looks reasonable. And what Archer also gives you is a whole bunch of peaks which go along with each of the different cell clusters. So these are different cell clusters that Archer gives, and then these are different peaks, lots of peaks that are associated with these different cell clusters. And you can look at these peaks, and you can see there are conserved peaks, and there are peaks that change across these different cell clusters. And so the key question is what are the genes that are associated that are associated with these genes peaks? So what does this peak actually regulate? And these peaks can be far away from the gene. So to look at that problem, we have developed another method which tries to leverage, I would say, fine clustering of single-cell attack-seq data to infer what we call as pseudoclusters. They are like metacells, if you have encountered that, but look at covariation patterns of accessibility and of a region that is far away up to a megabase away, and a gene, and then use that to say what might be a potential region that might be regulating a gene. So our overall workflow looks like this. We take a gene and look at a TSS, look at fit a regression model, a random forest-based regression model, and we figure out what are the different high confidence loops. And then we do some additional filtering based on Archer, and then and what we get is a ranking of now regions. So what we've done is first regress for each gene, and then rank regions based on the number of genes and the overall importance of of a region. And based on our different filtering criteria, we have now narrowed a set of regions that we think are interesting that we are now experimentally validating in the lab. So I'm going to tell you about one of these. And this is what we call this the FA1 locus for reasons because of lack of a better naming convention. But what you're seeing here are different tracks. And so this particular region is this particular one around this blue box. And this is interesting because it is associated with when the it's down in the MEF, that is the early time points, and then it becomes more up in the later when the cells have reprogrammed and become pluripotent. And what we've also done is also tried to see which of our peaks are dynamic. So using another method that I'll not get into, but what you're seeing here is how well, the pointer went away. Okay. What you see here is basically how well are we able to predict the accessibility of a gene based on this particular region. So these are two target genes, and this is showing the overall agreement between the accessibility of this region and the accessibility of this gene. And we see that it does a pretty good job. So these are like different target genes that we've predicted that we think are regulated by this element. And now what we are doing in terms of validation is using CRISPR-I to knock out or actually suppress this gene by using CRISPR-I, and we are measuring based on different modalities like K9ME3, extent of reprogramming, also target gene expression, and then seeing whether what we are getting actually makes sense. And what you see here is the overall reprogramming efficiency, and we see that basically when we knock down this particular locus, there is a significant increase in reprogramming. It has a significant effect in reprogramming, and it also affects the expression of these different genes when we have perturbed this particular locus. So at this level, it is able to pull out something that we think is making sense, but we are still trying to look at our other targets that we've predicted, other regions that we predicted that we would like to see how well they do. We are almost at, maybe we have like maybe 5 minutes or something.
We have plenty of questions.
Okay.
So, yeah, I would say go ahead.
Okay. So this is the last part is what we've spent a lot of time on is in terms of inferring transcription factor target relationships or this trans thing. And here, this is something that has been there for since when microarrays were there, and people have been applying methods out here. And now again with single cell RNA-seq data, people have been doing this where. What you're doing is you have this gene expression matrix where you have genes along the rows and you have cells along the columns, and you want to get out a GRN that connects regulators to target genes. And there have been a lot of reviews. You can check out these reviews, and also benchmarking studies, including one from our group where people have tried to compare gene expression, inference, single cell network inference methods to see which methods work well versus others. The problem is that most of these methods, again, here comes our trees, have been ignoring the fact that when you have single-cell RNA-seq data with multiple cell types that are related, they ignore this relatedness. And they have treated this as a single-task learning problem. And what we want to do is try to overcome that limitation, but also leverage accessibility information, if you have that, to infer these gene regulatory networks. So we've developed a method also using a multitask setting, but here the tasks are now network inferences rather than clustering. And it's called SCMTNI, and what this method does is it takes in a lineage structure, single-cell RNA-seq data for each of the cell clusters, and if you have also accessibility data, it can also take that, but that's not required, and then it outputs a gene regulatory network for each cell cluster. It uses the accessibility data to inform a prior. So if you have accessibility data, you can get information like which transcription factor binds to which locus in the genome, or if you have motifs for those transcription factors, and you can use that to provide an initial structure to the algorithm, and then we use that as priors together with the gene expression data to inform what the final network should be. Instead of using something like, I have this accessibility data, here is my GRN, I am just going to remove edges that are not supported by gene expression, we try to make it more relaxed in terms of incorporating gene expression. And the way we incorporate the lineage structure is by using a probabilistic prior. This is actually motivated by some work that we did when we were learning gene regulatory networks across species. So it's a very similar type of a prior, but it's different in the sense that cell lineages have data on the intermediate nodes, whereas in phylogenies, you don't have data in the intermediate nodes. So how does STMTNI perform? It this is based on simulations. Not a surprise, it works quite well on simulated data sets, but also on real data sets, it also works quite well compared to several existing state-of-the-art methods. But overall, what we found, at least from simulations, is multitask learning is a good way to infer gene regulatory networks across different methods. Sharing across different tasks is generally a good idea for looking at GRN inference. So now I'll tell you quickly about what we obtained in terms of applying this in a reprogramming setting. I'm going to skip this and just tell you that what we have is, so we have RNA-seq data and ATAC-seq data for this reprogramming system. And we used Liger to integrate the data. And what you are seeing here is basically a cell lineage structure that we inferred. So C4 is one of the cell clusters, which is largely made up of MEF cells. And these cells go along these two trajectories. One of them is becoming the pluripotent state, and the other one that doesn't reprogram, that is in the C2 state. So the question now is, what is it at the GRN level that actually drives these potentially? And so that's the question of defining dynamics. And here we've developed two approaches. One is very simple, based on edge clustering, which I'll not get into. And the other one is based on this idea of topic models. And this is actually, we've adopted this idea from another paper, but where people try to study regulatory network rewiring using this idea of latent durational allocation. In the interest of time, I'm going to skip that. But the thing that LDA, what this framework gives you, is it gives you groups of genes that we call as topics, and then you can look at those topics, and those topics look rewired. So just to give you an example, here is one of those topics, which is actually part of the network that we've inferred across the different cell clusters, and each of those dots are laid in the same way, but you can see that there are some, and the red lines here are the connections, regulators to target genes. You can see that some of the hubs actually are more apparent at some point in the cell cluster. So basically, this is zooming in into one of the into the C5 and the C2. These are the two endpoints of the reprogramming trajectory. And what we see is that in C5, we see a strong hub for ESRRB, and we see several cell cycle regulators. In the C2, we see that these hubs are actually not there. So that's potentially suggesting that the embryonic, that state, this component of the GRN needs to be upregulated. And more generally, we find more regulators. So these are different cell cycle regulators and ESRRB that needs to be upregulated for cells to reprogram. But then importantly, what we find is that regulators like that are associated with MEF tend to not go away in this other trajectory, which potentially makes these cells fail to reprogram or reach that final state. So these are regulators that we predict, that we think are responsible for the lack of these cells to not reprogram. We tried to test these, but it's still something that we are still working on. But these are hard experiments. Validations are hard experiments. But I mean, but what is nice is that, you're able to actually pull out these types of regulators. And we've identified these similar types of regulators in other systems as well. So, and many of them make biological sense. At least that's what our collaborators tell us. Okay, so that wraps up what I wanted to tell you. I'll just summarize the overall talk that I gave today, which is trying to infer gene regulatory networks from bulk and single cell data. And we have a lot of cool data sets, both from bulk and single cell omic technologies, but we need methods to try to look at what are the specifics about these data sets to get into gene regulation and regulatory networks. Validation still remains a hard problem, but with methods like Perturb-seq, perhaps we can try to do more high throughput validations and try to infer, to really evaluate how well our methods are doing. And with that, I want to thank my group and my collaborators, and I'm of course looking for postdocs and also grad students. If you have questions about my talk or anything else, I'm happy to answer in the last 5 minutes.
Any questions for Sushmita? We had several about the first half of the talk. All the single cell stuff is just so clear that.
Yeah, maybe the single cell stuff went too fast. Yes.
I guess I have a question. So for most of your talk, you've relied on having this network structure, this tree structure be known ahead of time. How much do you think you could, if you didn't have that, do you think you could reconstruct or leverage any sort of knowledge that there is a lineage, but you don't know what it is?
It's a very good question. And so we actually got that question for SCMT, and I, kind of got it, had to address that. So in the single cell, we are reconstructing the lineage from the data itself. And so we are assuming that we are doing a reasonable job of trajectory construction. So we are using the single cell RNA-seq data to get us pairwise distance matrix and then constructing the tree from that and then using it. So that would be one way to do it. But if you have, of course, known lineage structure, that can be incorporated. But you could incorporate that as part of the whole learning thing. But that's much harder. You could afford little variations, but largely the trajectory should be there. But you could, because for every trajectory, you relearn all the networks. But in theory, it can be done. But that's a great question. The other thing is to do an initial network inference and then look at similarities of the networks and then construct it from there and then refine the networks. So that would be another way. But I would say using gene expression, using an initial auxiliary data sets to try to inform the tree structure is a reasonable approach.
Go ahead.
How robust do you think these techniques are to missing information? So say you're working with non-model systems and you don't know if your genes are actually genes or if you've identified all the genes. Does it do enough leveraging across the different nodes to be able to accommodate that, or is that kind of through into things?
So I would say that the network inference is probably the least robust thing, and that's something that we know is a problem with single cell data. But I would say the clustering aspects that we've done would be a quite nice, we've developed methods to work with bulk data, and let's say that you're not sure whether it's a loss or whether it's just missing data. We still incorporate it into our framework, and we can, look at those results more carefully. So I would say that, when we are grouping things together, things will be more robust to noisy data compared to when we are working with individual genes, which is what we're doing with this GRN inference. Yeah, it's a good question, yeah.
I have a question, and then you mentioned about building cis versus trans GRNs, and as though building them separately. Yeah, what is the
Rationale.
Yeah, rationale, and then how would you combine them?
Yes, great question. So the reason why I separated the cis versus trans was because, so the cis, once you solve the cis, then you can actually incorporate it inside the trans thing. So let's say, so you do need the enhancers that regulate the gene's expression. So let's say that you solve that problem. So you know what regulatory elements, in addition to the promoter. So in our algorithm, we are looking at the promoter binding sites and then incorporating that as a way to inform which regulators expression level will change the expression level of a gene. But now let's say that you also brought in these distal elements. Then what you could do is scan them for transcription factors and then bring them into our overall model of learning regulators to target genes. So that would be one way to do it. It may not be very satisfying because some people say that you're treating gene expression as the final thing that you're producing. What about like trying to produce also accessibility as an output? But we haven't done that yet. So yeah, but that would be one way to use both of these things and to get the final GRN. Because ultimately you want to connect a transcription factor to a target gene. The question is, are we going just through the promoter? Are we going through the promoter and the enhancer to regulate that expression gene?
There was one more.
So when you use the single iron strip and single ATAC strip to pick out the PRNs, do you really use the predictive transcription factor binding motifs, the open chromatin region to
Do we use the transcription factor in the open chromatin to predict the GRN?
Yeah, like handle factor binding motif enrichment?
Yes. So the way we do this is like this. So I don't know what you mean by enrichment, but like what, so let's say that you called peaks, okay? So the way, if you have ataxic data, single cell ataxic data, you will infer clusters. For each cluster, you can call peaks by applying max, all given by Archer. So what you can do is then you can, and then separately, you can find motif instances, and these are available from Jasper. You can download these instances, or you could scan the genome using something like FIMO. So there are tools actually out there. Scan the sequence genome-wide. Then you can see whether the motif is under a peak. And then you can say, okay, so that transcription factor's motif is under a peak, so that transcription factor potentially regulates my gene. And that's how you would incorporate it inside the network inference.
All right, let us thank Dr. Roy.
That was awesome. Sorry, I gotta run. I've got another one at 1.
No
But that
problem.
was very
Yeah.
good.
Yeah.
There was a lot at the end there.
A lot of information. I should have just talked to us. The thing is,
Because this stuff was super interesting.
yeah, I gave a smaller version of the talk, but I added like the TMF because that talk got over in 35 minutes, and I was like, this is too short. What am I going to do with the rest of my time? So that's why I added this. But then in retrospect, I think in-person talks just take longer
Yeah,
because you
they
wait
do.
for
They
questions.
do. Well, no, and you got a lot of really good questions along the way.
Yeah.
Yeah.
I believe these might be your lunch days.
Oh, cool.
Cassian.
Hi, nice to meet you. Nice to meet you. Yeah,
I don't know what kind, sorry, I wasn't
nice to
paying
meet you.
We're both in Gene Robinson's lab.
Yeah.
And we're kind of both, or have been both, involved in different aspects of the informatics that you were just talking about.
Oh, great.
Cassie is up and coming with single cell.
Oh, cool. Exciting.
I'm looking at microbiome.
Yeah,
Oh,
so
cool.
I will preface this by saying I have very limited experience with actually analyzing sequencing data
Yeah,
as
that's-
of right now.
Yeah.
But yeah, so like I have all of my data, and I'm planning to kind of combine like metatranscriptomic data with data from honeybees to see if there's some kind of interaction between what the microbes are doing to the host.
Oh, that's
Sorry,
super
do
interesting.
you have a quick question before?
Yeah.
Hi.
So I was wondering how much work, if there's been any work on sort of spatial smoothness of the networks, right? So like we know, right? So like as a cell line is evolving, right, between sort of phenotypes.